and he waits and waits and waits and waits. And probably he won't make it because there's only about 10% of people who will get a transplant. There's a lot more people who want transplants than the organs available for them. Now what they've done in Wake Forest now is they're taking iPS cells and they're getting a scaffold. Now obviously if you take a dot matrix printer, right, you can print something out that's basically a 3D thing, and the little woodworking thing will cut a model for you, exactly what you put the program into, and there it is, right? You can't do a 3D thing of cells unless you've got a scaffold to build on. So what they're doing now is they're taking, let's take hearts, they're taking pig hearts. They wash all the cells off the pig heart, and what you're left with is a fibrous model, just the skeleton, the scaffold. Then you can take your skin cells, make iPS cells, push them down the pathway of becoming cardiomyocytes, heart cells, seed them onto the scaffold, the pig scaffold, and it will grow a heart. And all these little cells start to grow, and they start to beat. And they start to beat in unison. And you can watch them in a bioreactor. And they've done this. Admittedly a little tiny heart, because they're doing mouse and rat studies, but they can build a heart. And you think, wow, that's pretty spectacular. But what they've also done, and this is actually a clinical case, they have built bladders. People obviously need bladders, but they've built them and they've transplanted these bladders into people. 8 of them have gone in now, successfully. So you don't hear much about this stuff, but this is on the way, and it's very close to being able to. They've also made kidneys, little tiny kidneys, but you put them into the body and they filter urine. They start making urine in a little bag attached to a little kidney that's under your skin. So soon we'll have the ability to make your own personalised replacement tissues. And that's probably, if you get anything out of this, the take-home lesson. That's what's happening today. Okay, so let's talk about adult stem cells, because I work with adult stem cells. Now, adult stem cells occur in all tissues of the body. They are in the brain, in the skin, in the heart, you name it, they're there. If they're not there, we just haven't found them yet. They're almost sort of there, but they're in very small numbers. And their job, when the tissue is damaged, is to rapidly start producing new cells to replace the ones that are damaged. But, as I said, they're in small numbers. So, what we do is we take mesenchymal stem cells, which are adult stem cells, and we get them from a tissue that's in plentiful supply. We used to get them from the bone marrow, but now we get them from adipose tissue, of which we all have a lot of stem cells, or just fat. These mesenchymal stem cells actually have the ability, abilities are quite unique, and this is why we use them, because they're easy to get out of the fat, but they also have 3 properties that are not in any other stem cell. Number 1, they're easy to get to. That's the first thing. Number 2, they have the ability to avoid the immune system. So it goes against what I said originally, that I couldn't take yours and put them into me. I could take my mesenchymal stem cells and put them into you. But they don't have a great ability to change the different cell types. We can make them into fat, duh. We can make them into cartilage. We can make them into bone. We can now make them into neurons, and we can make them into heart cells. But not everything. So they're limited in their capacity to actually change, to differentiate. So mesenchymal stem cells, A, easy to get to, B, they avoid the immune system, and C, they are anti-inflammatory in nature, which makes them a really good cell type for my work, which is an osteoarthritis, which is what we do. Okay, so they're really the workhorse stem cell. Most of my patients are obviously dogs, and they've got a lot of fat, not as many as some. From this slide here you can see, this is an old slide, but you can see bone, fat, muscle, and cartilage, but there's also neurons and heart cells in there now. This slide actually shows you where the cells go to. I'm not a big fan of graphs, but this is a good one. When you put these cells into the body, they tend to track to the areas that are damaged. You can see that this is the lung area here. Most of these cells end up in the lungs. This is another problem we've got to overcome. If I stick them intravenously into you, obviously the lung tissue vessels get smaller and smaller and smaller as you get in a tree. And so eventually they get clogged up and they don't get through. but new research shows they do get through as they sort of weasel their way through after about 2 or 3 days. They do get there. But you can also see that the spleen, the pancreas, the kidney, the liver, muscles, brain and heart, they all get their fair share of these cells. So they do get there eventually. Okay, that just tells you that they should get there. This is a labeled cell picture of a heart muscle. This was a graft, what's called an allograft. It went from one animal to the other animal. It obviously wasn't going to take, the immune system destroyed it. But the little blue, can you see the blue there at all? Not a real good slide. Can you see blue? No? Okay. The little blue parts are the labeled cells, and they're actually accumulating in the tissue that's dying. Okay, so they're going to the damaged area. It just shows you that they migrate to the damaged areas. When you apply them locally, people ask me all the time, how do they do their work? Well, they do their work in one of 3 ways, but probably all 3 ways. Firstly, they go there and they differentiate in the tissues that are required, listening to the cell signals of the damaged tissue. Because obviously if you cut your hand, the next day it's red. And it's red because cells go there to kill the bugs that are in there and repair the damage. That's why it's red. Same thing applies to their stem cells. They go there, they listen to the signals, and they go, okay, we need to be turning ourselves into muscle or scar tissue or whatever. The other thing they do is when they get there, they secrete their own cellular signals, waking up the stem cells that are sitting there quietly, ready to go and do the job. And lastly, we believe that they merge with some of the cells and exchange body parts, for want of a better word. They exchange mitochondria and some cellular components, and all 3 together tend to do the job. That's what we think at the present stage. And when you apply them intravenously, they tend to home to certain tissues. This is a slide of a gamma camera scan, labelled cells with a radioisotope, and you track them. It's interesting for a couple of reasons. This gentleman had a disease called cerebellar ataxia, where he had difficulty in balancing. The cells were can you see that slide? Not really? Just. Okay, what you can see is there's black marks on it, right? So this is the bottom half, that's his head up there. These are lungs and liver up here. This is bladder as the cells get filtered out. This area here is his cerebellum. Now these cells knew the cerebellum was damaged and they accumulated in that area. Okay? This is them filtering out in the lung field and obviously you can filter in the bladder. The interesting thing as well is, you can't see this, but they're injected in this arm here. This guy also suffered from quite a seriously damaged right hand. They tracked there as well. Okay? So you can see that they actually go to the areas of damage. Okay. This is some of my patients. Yeah, this little guy obviously wasn't a patient, but fat's readily available in animals. I guess I can't retouch on obesity because there's an epidemic of obesity in people, as you would well know. But I got to say that there's an epidemic of obesity in pet dogs too. People overfeed their animals dramatically, so it's not unusual for us to see diseases like diabetes, arthritic change, animals that are obviously obese, and owners just can't see it. So for us, it's a good thing and a bad thing. For me, from the point of view of a stem cell person, it's great because I've got plenty of stem cells to work with. But on the other hand, being obese is not a really good plan. Let's talk about what I do now, which is osteoarthritic treatment with stem cells. Obviously, I'm dealing with a quadruped here. Everybody here is a biped, except for some Saturday nights when you're not really a biped. The arthritic change that we get is similar spots that you get. What are you pointing him out for? Oh, you. All right. Shoulders, elbows, stifles, coxofemoral joints, and in dogs, strangely enough, lumbar vertebra really get a bit of a hammering, depending on how active the dog is. You look at a 10-year-old Kelpie on X-ray that's had a life of chasing a ball or rounding up sheep or whatever, his body's really smashed. And you can see that from spondylitic change. Dogs have got 7 lumbar vertebrae as opposed to us with 5. And we see problems usually in that area and sometimes there. Dogs slope downhill, so each front leg carries 1/3 of the body weight. Each back leg carries 1/6 of the body weight. So often bigger, heavier dogs that have been performance animals like you know, Labrador retrievers have been working, you will get a lot of shoulder problems, a lot of elbow problems. All these problems respond quite well. Now, the look of osteoarthritic change in animals is the same as in people. These radiographs are disgusting. I mean, you wouldn't want knees like that. They look horrible. Instead of being nice and smooth, you can see all this fluffy osteophytic change. There's even joint mice floating around here somewhere. But they're really crap knees. You wouldn't want those knees. But that's pretty typical of a dog that's been, you know, working pretty hard all its life. And they're quite crippled up. This is actually, obviously, hips. The difference between this one and yours is it's got a tail here, so not a person. The hips, I don't think you can see it very well, but you can see, you know, spurring here on the acetabular rim here, and obviously these hips are partly out of the acetabulum, and they've got osteophyte formation all along that area. And they crunch and they grind when you push them around, and the dog's obviously very unhappy about that. Okay, let's have a look at the technique that we use to harvest these cells and how we process them in general terms. I'm not going to bore you with the laboratory stuff because that's not really my bag either. The cells are harvested, it's basically liposuction. You whack in a few digestive enzymes and you suck out all the fat and you end up with a whole heap of liquid fat, basically. Then we break it down in the lab with some enzymes to break the fat down, and then we centrifuge it out. The stem cells have a different specific gravity, and therefore they come in layers as you spin them down, and you can take them off there. And you take the stem cells out off that layer. Stem cells in fat are dormant, and they're just sleeping quietly there, and so what we do is we wake them up. We wake them up in two ways. We add what's called PRP, platelet-rich plasma. If you go to the cosmetic surgeon to get some work done on your face, and there's some candidates here this morning, then they'll often use PRP. PRP is platelet-rich plasma. Now, platelets, which are obviously the clotting cells of your body, they contain about 400 different factors inside them. And when you release these factors, they stimulate stem cells, which is why people like Madonna, they tell me, get this stuff injected into their face. So it's quite common for cosmetic surgeons to use PRP. We use PRP, which we take from the blood, to wake up stem cells. We also wake them up using lasers. For some unknown reason, stem cells like the colour yellow and the colour red and the colour green. They don't like the colour blue. Blue puts them to sleep, the other three wake them up. So the lasers we use are a multicolour, those three colours laser. And we irradiate the cells for about 30 minutes, and it tends to activate them or upregulate them. And then we wash them and off we go. These are they. Strangely enough, these cells are little tiny ones with a little black nucleus. When you actually grow these cells up in the lab, they don't look like that. They actually look like that. If you can see the ones on the side here, like a spindle shape. These are typical fibroblast-like cells. These ones here are usually stem cells, or what they call progenitor cells. And there's lots of them. We actually harvest approximately 3 to 4 million cells per gram of fat. And proudly he says the US are only getting 300,000 cells per gram of fat. So, yay us. They don't like us very much. Okay, so let's have a look at how stem cells are currently being used. Lots of different techniques being tried. Mastectomies, they've applied stem cells here, often in the fat, to regenerate breast tissue. They're actually in Asia using it for breast enlargement. This young lady was badly damaged in a landmine, and you can see the difference that made to her in facial reconstruction. I take these before and after pictures always with a grain of salt, because you often see the after picture is well made up and lippy and the rest of it. Hair growth. You can see that the follicle stimulation from injecting these cells is quite substantial. It's after 10 weeks. This is the same one, and I kept this slide just because I wanted to show that guy.